Now we have the eukaryotic cell. The eukaryotic cell, for all of its majesticness, is a membrane-bound nucleus. Obviously, that was the thing that we just talked about being a major difference between us and the pros. We have a vast number of organelles, okay? A lot of them, all right? A lot of them. You don't see a lot of this specialization in bacteria. Okay? We have an extensive plasma membrane, even more so than the prokaryotic organism, all right? And this plasma membrane is really this barrier, this boundary that keeps things out and keeps things in. And it only lets things across that it deems necessary. Okay? The first two are what distinguish us from a prokaryotic organism. Nuclear-bound nucleus, okay, membrane-bound nucleus, and a vast number of specialized organs, okay, called organelles. That just means tiny organs. I mean, eukaryotic cells are bigger than prokaryotic cells. I don't know if that's going to take you an earth-shattering amount of time to memorize, but I'm not going to ask the distance, diameters, or anything like that. But they are bigger. Okay. Yep. Organelles, specific functions, and they're tiny. Okay. That's what you're going to have to know. Functions for all of them here. There is this thing referred to as an endosymbiotic theory. It's a hypothesis really on how the eukaryotic organism came to be. And there's a little video that's on the next slide here, and I'll let you watch that on your own. I want to just kind of keep going here. But we have a number of various different biochemical data points to suggest this theory, looking at how we got a membrane-bound nucleus, okay? How we've got mitochondria, chloroplast, energy-producing cells. It's those energy-producing organelles that we have inside of us now that allow our cells to make a vast amount of ATP to do a lot of work. So, we can get big, okay? We can have a lot of specialized now tissues and organs because we have the ability to take those biomolecules, make energy, and now I can make some very complex structures because now I have the funds to support their functioning. And that's all it talks about here. Again, here's that video here. I'm going to keep going because I'll start playing here. But here's kind of what I'm talking about here. Here is just this original kind of prokaryotic cell. Original prokaryotic cell, all right? Then it started to kind of fold in on itself. And I hope I can get that video to play a little bit later here in the semester when we start looking at evolution, because we'll see how, or the potential of how all of these adaptations kind of began. And it's really blind luck a lot of times, but there's some other things that go into it. So if we can watch a little bit of that video, it's really long, but only really the first like 45 minutes to an hour are really any good. But it looks at how these little adaptations happened and occurred. And it's just lucky that we're here when you watch this video, like, holy crap. So here you can see that invagination that's kind of occurring right here and how we get that kind of nuclear membrane to kind of take shape and take hold and really separate us from a eukaryotic cell. Then we're kind of this proto-eukaryotic cell, and then all of a sudden we have these aerobic bacteriums, mitochondria. They take up residence inside of our ancestors, if you will, of a eukaryotic cell, this proto-eukaryotic cell. Mitochondria, chloroplast. And we really start to see this cell take on a lot of specializations to where it starts to, oh, chloroplast went in this cell and these cells divided, okay, it's going to go this route and it's going to become a plant, okay? No mitochondria went inside this cell and this cell replicated and went this way and became an animal cell. right? And that's kind of the origin of all these organelles, because now we have some specialization. We got some pinching in of that plasma membrane, that phospholipid bilayer. So now we're going to have this thing called an endomembrane system. That plasma membrane folded in and started to give rise to various different numbers of organelles. And when we talk about that, that's what the endomembrane system is. All it is membrane-bound organelles. That's all I'm trying to say here. All right. This is the nuclear membrane, the nucleus. This is the vast majority of organelles, except 3. Mitochondria, chloroplast, and ribosomes. They are the only things that are not really associated with this endomembrane system. All right. The endomembrane system is just a series of various different organelles that all communicate with each other through tiny little vesicles. Okay. They're just, think of these as a dump truck. Okay. Tiny dump trucks. Tiny dump trucks. Okay. T-R-U-C-K-S. I know you had to spell trucks. But these little tiny vesicles just transport stuff around the cell. Oh, this guy needs it? Hold on, package it up right here and I'll take it over here. Oh, you need some over there? Okay, I'll package it up and I'll take it over there that way. All right. And I hope I have a video. If I don't have that video, I'll find it and show you, because you can see how all this stuff that we're going to be talking about interacts with one another. It's weird. It's crazy. The energy-related organelles are mitochondria and chloroplast. They are independent, man. They are their own man and woman. All right? They are self-sufficient. They have their own DNA. They have their own ribosomes. They are their own entity. That's why we have this little, the proto-mitochondria, this aerobic bacterium, making energy. It's like, oh, man, I'm getting targeted. I'm going to go inside this cell, and it's going to protect me, and in the process, I'm going to give it ATP. All right? So that's why these guys are their own person, mitochondria and chloroplast. Here's what a typical cell looks like, or an animal cell, I guess I should say, looks like. Okay, we're going to be talking about all this stuff. You're going to be able to know exactly what all these things do if they're located in a specific region, all right? But it looks like a lot, but there's not. There's not a whole hell of a lot of stuff at all, all right? But that's a typical animal cell, okay? It could maybe show that on the exam, maybe have you identify some things. That would make a good question. Here's a plant cell. It says plant cell, so that's how you know. And here you can see it has that kind of rigid cell wall around it here that helps to kind of give it its kind of uniqueness. That and those green things called chloroplast, and yes, they are really green. Okay. But as you can see, I mean, all the same stuff. Nuclear envelope, nucleolus, chromatin, nuclear pore. I mean, all of these things, sans, cell wall, and chloroplast are found here, right? So it's not like you're memorizing two way different things at all. You memorize it once, you got it in both, except cell wall and chloroplast. Okay. What's on this thing? What's on the next slide? Okay, I just talked about it in the membrane system, everything being interconnected by the vesicles. Yes. We're going to talk about, making things, making proteins, making fats, how we transport those around, and just how they all how they move those things around. Because it may seem like, oh, it's just going to, it just floats. It doesn't just float, okay? It's very regulated, just like anything else in life, right? All right. So let's jump into this thing, all right? Let's start with the nucleus. Here's what it looks like, all right? Here's the animal cell here. I've plucked it out, and there she blows, okay? So there is your nucleus. It's got a nuclear membrane. Fancy pants call it nuclear envelope, okay? But nuclear membrane, nuclear envelope, it is the same thing. You can see it's dotted with little holes, okay? That's the nuclear pore. you can see it's got squiggly lines in here. That is the genetic material. This right here is what we call it, chromatin. And it has, because it is separated, it is a defined structure by this nuclear envelope. Okay, it has its own cytoplasm inside of it, and we ingeniously call that nucleoplasm. So let's go back here. The nucleus, what does it do? Well, that's pretty easy, right? It's the command center of the cell. It's usually in the center, doesn't have to be, okay? Different types of cells, different types of tissue, they can be in different locations, but it is the command center of the cell. It is the brains for the cell, okay? The nucleus is sending out information, telling that cell kind of what to do. It does that in the form of proteins, all right? It is defined. It's separated from the cytoplasm that is the fluid, inside the cell itself. And that's that nuclear membrane. It's a phospholipid bilayer, just like the plasma membrane, okay? It's just defining the shape of the nucleus, so they call it a different name, nuclear envelope. Again, little holes dot this nuclear envelope, all right? And this is how things get out of the nucleus. And you're like, what in the world's coming out of that thing? DNA, okay? Well, specifically RNA, all right? It's coming out of that thing. We have, again, chromatin that are within that nucleus. And that semi-fluid content of it is, there it is, nucleoplasm, okay? I should have highlighted this. I don't know why this made it past the goalie, okay? But we also have a dark circular structure within the dark circular structure, okay? And that's this thing right here. And that's called the nucleolus. And the nucleolus does one thing and does one thing only, and it makes ribosomes, okay? Makes ribosomes. And ribosomes are complex. There are two parts to it. That's why I say subunits. It'll make more sense when we look at ribosomes here in a couple slides from now. So the nucleus, what does it do? Command center of the cell. What does it have? Well, it has a nuclear envelope, okay, a phospholipid bilayer. It has cytoplasm inside of it called nucleoplasm. In the surface of the nuclear envelope are tiny little holes called pores, nuclear pores, because it's on the nucleus. The nuclear pores allow ribosomal subunits and then copied genetic material to leave. Okay, I think I have that maybe on another slide, because I don't have a lot of space to write here. Okay. So that's what we have for the nucleus. Those are all the little parts and pieces that we have so far. Scanning electron microscope, you can see what that nucleus kind of looks like. Really unspectacular, right? Ribosomes, I don't really have, well, there's a picture a little later here, but it's comprised of two subunits, okay? A large and a small. Okay. You put those two together, and that is the ribosome, right there. Okay. Ribosomes are just basically composed of RNA. When I put that R in front of RNA, because you have a lot of types of RNA, you have rRNA, messenger RNA, transfer RNA, mitochondrial RNA. So you got a lot of RNA out there. Okay. So when I put the small little R in front of RNA, okay, that's telling you this is ribosomal RNA. And that's what ribosomes are made of. Shocking. Okay. Again, subunits are made inside the nucleolus. Ribosomes leave through that nuclear pore, okay? Exit the nucleus, go out into the cytoplasm. They can roam around free out there, okay? They can be by themselves, float around. They can be clumped together in a whole mass of them, kind of moving around as a herd. They can also be attached to things. And they will only attach to this thing right here called an endoplasmic reticulum. So ribosomes, once they leave home, they can go anywhere. They can live by themselves in an apartment. They can live in a herd and travel around in a big pack. Or they can go and attach to another structure and make their home there. All right? And that's called the endoplasmic reticulum. It does not matter where it's found, by itself, in a mass, attached to the ER. The function is always the same, and FX is shorthand for function. It is the site of protein synthesis. Site of protein synthesis. Every time, okay? By itself, in a group, attached to the ER, anytime, all the time. Its function is always protein synthesis. So if we see rough endoplasmic reticulum, like we will here in a little bit, and I'm going to ask you the function, as soon as you see rough is telling you ribosomes are attached. And if ribosomes are attached, it's always involved with protein synthesis. Okay? If it's not attached, it does something else. Okay? If there's no ribosomes attached to that endoplasmic reticulum, then it does something completely different. Ribosomes are involved with protein synthesis. What are proteins? Proteins are made of what tiny structures? What's their monomer? Hopefully you are sitting there at home going, amino acid. Amino acids are individual and they get strung together by DNA. Okay? DNA is the blueprint for how these amino acids are going to get stuck together, right? So ribosomes are going to be involved in this process called transcription and translation. Okay? This is where we copy DNA. Copy DNA. Okay? This is where we read that copy. Read the copy. Okay? If you remember that part right now of transcription and translation, I will be happier than a pig in poop. Okay? Because we're going to get into a lot, well, not a lot more, but we're going to get into a heck of a lot more than just saying copy of DNA and read the copy of DNA. All right? We're not going to get into all the super detail that the book does, but we're going to get into enough detail that we can feel comfortable that we can know what this is, and we can, talk to somebody at a party next time what transcription and translation is. But the ribosomes do this. Okay? The ribosomes do this. The ribosomes will take that ribosomal subunit from the nucleus, right? Will take that copy of DNA, okay, that's been transcribed. It takes it out into the cytoplasm, squirts it out there. Okay? We have one subunit attached to that mRNA strand, messenger RNA, and the other subunit comes and attaches to it, and then it travels around it, and it reads it. When it reads it, we call that a translation, right? When you read something and translate it, and say you're reading a book in Mandarin, and you want to translate it into English, that's what you do. You're reading something and you're making a copy in something that now can do something, all right? This is that central dogma that I was talking about. DNA to RNA to proteins. Okay? And now proteins can do stuff. They can make structure. Proteins can make structure. structure. Okay? Ribosomes are central to this process of the central dogma. So if we go here to this slide here you can see, here's my ribosome escorting my messenger RNA out of the nucleus. Okay. Here you can see it's reading it. We're translating the sucker. Okay? It can stay out there by itself. We can dock to the rough endoplasmic reticulum. Okay? We can do a bunch of different things with that. Again, we'll get into this a heck of a lot more later. But that's what ribosomes do, make proteins. Okay? They read the messenger RNA, string together amino acids, and now I have the end result, and that's protein. So let's move out of the nucleus and enter into this thing that we're going to be talking about here, this endomembrane system. What is this? It's really all of the other organelles outside of the nucleus, right? The nucleus is kind of the hub, the beginning of it all, and then we extend out and go from there. ER. I keep saying ER, okay? Endoplasmic reticulum. The Golgi apparatus, and a vast number of vesicles here. So let's dive into it here. So here is my endoplasmic reticulum. All it is, guys, is a number of membrane channels, okay? Tiny little sacs. And tiny little sacs are called saccules, all right? So this flattened bunch of membrane channels basically, guys, extend from that nuclear envelope all the way out, potentially, to the plasma membrane. All right, that is something that's weird. That is something that's different. That is something that's unique. Unique pretty much gets asked on an exam. All right, so ER is weird. All right, ER extends from that nuclear membrane, and it may extend all the way out to the plasma membrane. All right, it may not go all the way out there and touch it. Okay, but it extends out a heck of a lot longer way than what you would think. As you can kind of see from the picture here, we have two types, 1 and 2. Okay, rough endoplasmic reticulum, which I have shorthanded ER, okay, and smooth endoplasmic reticulum. Again, you can use these abbreviations, just spell it once for me, please. That way I know that you know what those things mean. The difference between a smooth ER and a rough ER is one thing, ribosomes. Rough ER has them on its surface. Smooth ER has the NO version of that. Okay, no ribosomes. Because I have ribosomes attached, what do? Okay, we make proteins. Okay, that's what it means. Protein anabolism, anabolic steroids build. Okay, protein anabolism, synthesizing proteins. Why didn't I just put that? Because I'm weird. All right. So this thing will make proteins. It can modify the proteins. It can process that protein. What the heck does that mean? Oh, it'll add stuff to it. It'll maybe cleave stuff away and remove it and get rid of that. Now I'm the functioning protein. All right. It packages up this protein or modified protein into a vesicle. All right. And that vesicle will take it to, well, it could take it to the Golgi body, or Golgi apparatus is the name this book uses, which is fine. Okay, or it can get rid of it, send it outside of the cell. Or hell, it can stay inside the cell somewhere. All right, so it can go in a multitude of ways. But I package it up in that vesicle for transport around the cell. Smooth ER is the opposite. This is involved with synthesis of lipids. Now, smooth ER is highly varied. Okay, rough ER is rough ER. Okay, protein synthesis all the time. If you have a cell that's located in the testes, then, or the ovaries even, you'll make testosterone and estrogen respectively, all right? So here is the example I've written there. Smooth ER is adapted in the testes, so the cells that are within the testes, to help make testosterone. Smooth ER that we find within liver cells are more adapted to detoxify, process, and store lipids and other things, okay? But it stores a lot of lipids, all right? If it's inside some other cells, smooth ER will be adapted to store things there as well, Okay, so, and not detoxify and not process, okay, but just store stuff. So smooth ER can vary quite a bit on its function. It just depends on where the cell is located, in what tissue, in what organ. So this one, location, location, location means a hell of a lot more, all right? Same thing, package it up into a transport vesicle for really to go to the Golgi body or anywhere else inside the cell, just like it did with rough ER, right? So in that respect, they're both the same, but their function, way different. Rough ER, protein synthesis, smooth ER, lipid synthesis. So here you can see we transport, there's a little transport vesicle here, okay? It leaves the endoplasmic reticulum, whether it's smooth or rough, doesn't matter, okay? And it moves to the stack of pancakes. And to me, that's what the Golgi apparatus looks like, just a stack of pancakes, okay? Flattened saccules. And here is where we process and package and modify proteins and lipids. Oops, wrong way, okay? There's a cis and there's a trans phase. I'm not going to ask you, okay? Not going to ask you, all right? But Golgi body, what does it do? It modifies proteins and lipids. What does that mean? It may add a little something. It may take away a little something. It fine-tunes the product for final use, okay? And hopefully I have that video. If not, we'll find it. And we'll kind of see a little bit of that process. It doesn't get into the weeds of all of it. Again, once it passes through that stack of flattened pancakes, okay, that protein or lipid, it packages it up into a vesicle and then it goes to wherever it's going to go. Generally, out of the cell. Okay. See, cis and trans phase, I'm not going to ask you that. So don't worry about knowing cis and trans phase. Okay. But again, it passes through. I feel like I got to sneeze. Passes through here, packages it up in a little bitty vesicle. Excuse me. Packages it up into a vesicle for then removal from the cell. Now, that picture is a little bit disingenuous with up here. Okay, by the look of that exocytosis, you would think that all that's waste. And a vast majority of times it is not. May have some, but usually it's going to make some things that the cell needs. Lysosome, break the word down. In biology, break the word down into its component parts. Okay. Some means body. Lyse means to destroy, to chew up, to destroy. Okay. This is a cellular body that contains digestive enzymes. Okay. Produced by the Golgi body, whatever. Okay. Excuse me. But what's inside this cellular body? Okay, so means body. What's inside this thing? Digestive enzymes. Okay, this thing can do a number of different things, right? It can travel around the cell and help to recycle cellular resources. What does that mean? If there's an organelle that's not functioning, this lysosome will be told, hey, this guy's not working properly. Go attach to him and kill him. So it'll go and attach to it, dump its digestive enzymes into the organelle and destroy it. And then the cell takes those little parts and pieces and ta-da, remakes them into something else. It can also engulf, okay? If the cell was a white blood cell, okay, a leukocyte. White blood cells fight off foreign invaders, all right? White blood cells, they have a number of these lysosomes within them. So the white blood cell engulfs it and then shoots it up with all of its digestive enzymes and destroys this foreign invader, this pathogen. Here you go. That's what it looks like. Okay. Swallowing up mitochondria that ain't doing its job. All right. So here I've kind of, talked about this whole little endomembrane system, okay? Nuclear membrane, nucleus, nuclear membrane, endoplasmic reticulum, rough or smooth, vesicles, Golgi body, okay? There you go. See, from the nucleus out to the cytoplasm, where I read that DNA, or I incorporate lipids, okay? And that comes from someplace else. We'll talk about that later. Incorporate that in here, okay? Take it into the smooth ER, modify that lipid, take that protein to the rough ER, modify that protein. Take it to the Golgi apparatus, sorry. Okay, process, package, fine tune our final product, and then keep it inside the cell, do something there, or get rid of it, okay? Excuse me. I have two other major cellular bodies, okay? These are very specialized, as you can kind of see from the title there. They can house some specific stuff. Really, the biggest one is going to be a peroxisome, really, inside of us, animal eukaryotic cells. This carries peroxides, okay? It helps us to break down, catalyze hydrogen peroxide, okay? Peroxisomes, cellular body that contains peroxides. So this is an enzyme that's going to help us to break down hydrogen peroxide. This is just a byproduct of some metabolic pathways, okay? Hydrogen peroxide by itself is very detrimental to cells, okay? Very detrimental to cells. So I have this enzyme in the peroxisome that helps to break that down so the cell doesn't get hurt anymore. That's what peroxisomes do, okay? You see them in lipid breakdown or lipid metabolism, sorry. That's where you would see a lot of these. So in cells where you're going to have this thing called beta oxidation, which is using fats to make energy, you're going to see a lot more peroxisomes inside those type of cells than cells that don't use lipids for energy production. There it is, okay? Dyed yellow for your pleasure. The last thing we have here are what are called vacuoles, okay? Before we get to the energy producers here, are vacuoles. And vacuoles are much larger than a vesicle, okay? Vesicles are small. They just transport small units of stuff around a cell. A vacuole is big, okay? It stores stuff. This is its function, stores stuff, okay? So you typically see these in plants, all right? This thing is called a large central vacuole, and it's large. Here's a picture of it. There you go. Takes up the whole dang cell almost, okay? Large central vacuole. 90% of the volume. I doubt I would ask you the percentage, but there's to impress upon you how big this thing is, all right? This large central vacuole inside of a plant stores everything: water, nutrients, pigments, waste products, everything. This is how plants, when it's dry outside, this is how it gets its water to stay alive. Okay? It'll take it from here, all right? The presence of this large central vacuole stuffed to the brim with extra crap is also how this plant stays upright, okay? Again, if there's not a lot of water, okay, like there have been here except for the last few days, this is why plants start to wilt, okay? Because it sucks that water, it takes that water from that large central vacuole and it gets smaller and smaller and smaller. So now it's not putting up any pressure against the cell wall of that cell. And it goes meh, and it wilts. Okay? We have vacuoles in eukaryotic organisms as well. It's called a contractile vacuole. You'll see it in some basic paramecium-like structures, some basic eukaryotic organisms, and it helps them to stay alive in waters that change salinity, change their concentration. Okay. We have our two energy producers, and we were going to be looking at these ad nauseum a lot more in depth in the next unit. Okay, when we talk about photosynthesis and when we talk about cellular respiration. All right. But we have our two energy producers. They are the chloroplast and they are the mitochondria. All right. They are both their own entity. They have their own genetic material. They have their own DNA, like genetic material. They are their own entity, own proteins. They do their own work. They are double membrane bound, just like all the other organelles we've talked about for the most part, except for ribosomes. Because they are their own thing, we have some slightly unique names for them. We have this inner membrane fold, okay? There it is, okay? There you can kind of see it, okay? We have an outer membrane and an inner membrane, okay? And it's filled with these little, basically, stack of coins, okay? These little stack of coins are individually called thylakoids. Okay. When there's a number of these individual coins stacked, we use the word grana. Okay. Individual, stack of the individuals. Okay. It is within those structures, those thylakoid membranes, that we are going to undergo photosynthesis. And that's why these names are important, because we'll expand upon them in the next unit. Okay. They are green because of the photosynthetic pigment chlorophyll. Okay, it is a green pigment. All right, so that's why these things do appear green. Okay, again, what's a chloroplast function? To make energy. It does that by capturing sunlight and going through a process called photosynthesis. What is photosynthesis? Really simplistically, it is on this slide. Okay. It needs sunlight. It needs water. Okay. It needs a carbon source, carbon dioxide. Okay. As long as we have those three things, okay, as long as we have sunlight, water, and CO2, I can make, okay, carbs. And these carbs, sorry, I keep stepping on my shoelace. And these carbs help us to make energy. ATP. Okay. So photosynthesis is basically this. Okay. How it captures that sunlight are through those photosynthetic pigments. How does it get CO2? Because of us. Exhale our product of CO2, our waste product of CO2. It takes it in through the little holes on the underside of the leaf. Where's water coming from? The ground. As long as we have those three parts of the cocktail here, it can make carbohydrates, which allow plants to do a multitude of things. Mitochondria. The biggest thing that's probably missed on an exam or confused on an exam is where this sucker is found. It is found in everybody. Okay? Animal, plant, and even the bored algae. Okay? People always associate the mitochondria just with the animal cell. It is not with the animal cell. Okay? It is also found in the plant. Okay? It is also found in the plant. And we'll look at the process when we look at the next unit. Okay? Smaller than a chloroplast, meh. Okay, again, own ribosomes, own DNA. Okay, it makes energy. Okay, that's its function. Okay, it makes energy. Does this look familiar? Double membrane? Yes. Okay, it has two membranes. Yeah, an outer membrane and an inner membrane. Boring. Okay, just like the chloroplast did. Chloroplast has its own name. It's called its own little space here filled with fluid. It's called a stroma. I didn't have it on the previous slide, but I'll just mention it now. Okay. Ding dong. All right. Here, it has its own kind of centrally filled fluid region, and that's called the matrix. So stroma in a chloroplast, matrix in the mitochondria. Okay. Just cytoplasm-like material. It has folds in here. In the in the chloroplast, we refer to these as the thylakoid, as the grana. Okay, these two right here. In the mitochondria, we refer to these specializations inside this matrix as cristae. They're just folds. That's all they are. Okay, what do folds do? What do folds always do? Folds increase surface area. Okay, always do. What do mitochondria do? You know it, right? Powerhouse of the cell, okay? But let's not say that, all right? Even though that is what it is, right? It produces ATP, a lot of it, a buttload of it, a technical term. This making of ATP, of energy, right? Energy, is called cellular respiration, okay? This is where we utilize oxygen to make a buttload of ATP. That's what the mitochondria does. Chloroplast uses sunlight, carbon dioxide, water. Mitochondria, I need a carbohydrate source or some other sources, but carbohydrate's the primary energy provider of the body. I can use that carbohydrate and I can use oxygen and I can make a whole mess of ATP. Right? Without oxygen, I can't make a whole mess of ATP. Okay? And I can't do as much work. So that oxygen being present there is a pretty important concept when it comes to the buttload of ATP that the mitochondria can make. Okay? So you can see, sun, chloroplast uses that, CO2, it's water, and it can make a carbohydrate. You can see carbohydrate can do a number of things in a plant cell. But what does a mitochondria love to utilize to make energy? Carbohydrates. So it takes that carbohydrate, use it to make energy. Papa, ATP. And the process gives off CO2 as a waste and water as a waste. Oh my gosh, what does the chloroplast use? The waste. Put them back together and I can make a carbohydrate. And the circle of life continues. This is fun stuff, right? I could talk for hours on end. Okay. Do you guys want to take a break or do you want to keep going?